Hello students, today we are going to study the topic of PCR. PCR stands for Polymerase Chain Reaction. Okay, the full form for PCR is Polymerase Chain Reaction. What does it mainly use for? It is used to amplify. Amplify means to increase the number of copies. Okay, it is used to amplify the amount of DNA. For example, suppose you have only a very small piece of DNA and you want to make a lot of copies of that DNA, for some research purpose or some other purpose. At that point of time, you need the same copy of DNA in a larger number. So the PCR is used to amplify, to increase the number of the amount of DNA that you have, so that you can work with it more easily. Also, PCR is a tool for our DNA, recombinant DNA. Now, there are some steps that we need to follow when we are going to do this PCR cycles. The first step is called denaturation. What is denaturation? It is to separate both the strands of DNA. So both the strands of DNA will be like this. What will denaturation do? It will break the hydrogen bond that is between the two strands, and it will make sure the two strands are separated. How is it separated? By applying a high temperature to it. What is the temperature? It is 94 degrees Celsius. So at high temperature, the hydrogen bonds, they break, and the two strands of DNA are now separated. Now both the strands, they act as a template strand. The second step is called annealing. Annealing is the method in which we apply a primer at the 3' ends. This is important. We have to apply the primer at the 3' end. So with annealing, what happens? The primer is added. Now what is a primer? It is a RNA strand, single RNA strand. And this primer is 6 to 10 base pair long. Here we had 94 degree in denaturation. For annealing, the temperature comes down to 60 degree. So after denaturation, that means separating both the strand, a primer is added at the 3' end so that extension can be done. The third step is known as extension. Now what happens in extension? First of all, we need to remember there are 3 important properties we need for extension. One is called a Taq polymerase, second is a DNA nucleotide, and third is 72 degree Celsius. Very, very important. So for extension, these 3 qualities has to be fulfilled. So Taq polymerase, DNA nucleotide, and 72 degree. What happens over here? Simply, along the 3' end, new nucleotides are added, and as we have studied earlier, always the extension is towards the 5' end. Always remember this, that new RNA or DNA strand always get extended from 5' to 3' end. This is very, very important. Always the new DNA strand gets extended from its 5' to its 3' end. So these are the things that we need for the extension of the DNA. Now, why do we use Taq polymerase? Why not do we use the DNA polymerase that we use in our body? As we can see, the temperature needed for extension is 72 degree Celsius. DNA polymerase acts best at 37 degree, which is our body temperature. But here we need a higher temperature. Now DNA polymerase will denature at such a higher temperature. So we need Taq polymerase. Taq polymerase is a thermostable or a thermoliable enzyme, and it is acquired from a organism called Thermus aquaticus. This is a very, very important topic. Please make sure you mark it. Taq polymerase is acquired from an organism known as Thermus aquaticus, and it is known as a thermostable or a thermoliable enzyme. It can work very nicely and very efficiently at 72 degrees Celsius. So this is the reaction of PCR. There are 3 steps. The first step is denaturation at 94 degree. Second step is annealing, where primers are added at the 3' end. And the third step is extension, where new DNA strand is made from the 3' to the 5' end. And the things required are Taq polymerase, DNA nucleotides, and 72 degrees Celsius. Now, another important thing we need to remember is, in one cycle of PCR, 2 DNA molecules are produced, or in one cycle of PCR, 2 DNA strands are produced. We apply or we run the cycle of PCR for 30 cycles, so that 1 billion copies of the DNA molecules can be produced. Now, what is the formula for this? The formula is 2 to the power n, where n is the number of cycles. For example, if we run the PCR for 8 cycles, the DNA molecules will be 2 to the power 8. Okay, 2 to the power 8. number of DNA molecules will be produced. So this is all about the PCR. Thank you, students. See you in the next video.